In this experiment we're going to have a look at the extraction of DNA from some fruit. I've got here some strawberries, but I could equally well do it with a banana, and this is an experiment you can easily do at home. What we want are some strawberries. Now I've taken off the green bit simply because it makes life easier, and I'm going to put these into a Ziploc bag and I'm going to close it, and then the fun bit starts. What we're going to do is we're going to crush these strawberries, and we need to take a while here because what we're going to try and do is break up the cells, and by squashing them this is going to break all the cells, and it's going to release the DNA from the nucleus of these strawberries. So I want to try and get rid of all the big bits, and this is going to take a little while to mash up, but what we want to do is spend some time trying to get this really nice and pulverized. Fresh strawberries work really well. These were hard to get because this is winter time here, so not as easy to get, so these aren't the best strawberries to break up, but they still have all that lovely DNA. So while I do that, the next thing I need to do is to try and break this up even better, and we need a special solution to do this. So I'll leave that to the side for a minute and make up my solution. For this solution, what we want is some real salt. It's best to use sea salt. We don't really want salt with iodine in it, and what I'm going to do is I'm going to pour some water. We want 180 mils of water, and I'm going to pour some of that in here, and then because this is a laboratory rather than a spoon, I've got a stirring rod, and what I want to try and do is get all this salt to dissolve, and it doesn't take very long to do this, and we can get it to dissolve reasonably nicely, even though there are quite big crystals in here. So I've put in about a large spoonful of this, teaspoonful, so that's all we really needed to try and get it going, and that's dissolved nicely. And now what I want to do is I've got 90 mils of water in here. I'm going to add 20 mils of just washing up liquid, and I'm just going to pour this in. I don't want to get too many bubbles, so I've got this up to about 110, and now I can pour that mixture in here, making a lovely sort of yellow colour. If you can get clear washing up liquid, it's better for this experiment. It's easier to see, but it works just as well with any colour. So there is our solution that we can now mix up. Now I don't really want many bubbles, so I want to try and avoid the bubbles if I can. I've got some, and we can't go without any, and that's fine. So I've mashed this up quite well, these strawberries, and so now what I want to do is, having broken the cell walls up as much as I can, I want to try and break up the cell membranes, and that's what the detergent is for. So let's just pour this in, and it makes a lovely liquid. There we are. I'm going to close the Ziploc bag again. That's trying to stop all this going anywhere. And now what I want to do is give it a good mix, trying to get rid of all this DNA out of the nucleus into the solution. And basically we give it good sort of squeezes, trying to sort of mix all of this lovely DNA with the liquid, and I'm trying to get the DNA out of the cells into the liquid. You'll see that I've got quite a few bubbles here, but you want to try and minimize those bubbles as much as you can, because what you want to do is you want to get the detergent to be extracting out, destroying those cell membranes, so that what we've got is all the contents of the cell coming out. So I'm going to give this a mix for about a minute or two, trying to get everything out. Now, when I've done this, then what we need to do is to get rid of all the solid material, because hopefully all the DNA that we want is in the liquid. Now, when people say, I've seen that done before, to try and get this out, the best thing to sieve with first is a sieve. This is quite a coarse one, and what I just want to do is get rid of all the big lumps of strawberry and just get the liquid. And then when I've done that, I'm going to filter it. So I'm hoping that that's not too bad. It's not that lumpy, so let's have a go at doing the next stage. So what I want to do now is, I've got my bag there. I'm going to get my other one that I used. It's got some solution in it, doesn't matter. And what we're going to do is we're now going to filter this through this sieve and collect this liquid that we want. So now let's try and pour this in. Quite a messy operation, and you can see that I've come prepared for that because I'm doing my work on a spill tray. Let's move that one out the way, and there's all my lovely liquid coming out. And if I want to, I can try to sort of get a bit more liquid out through here just by mixing this round a little bit. And there we've got our lovely mixture of all the strawberries, that basically the stuff that hasn't come out. So what I'm going to do is I'm now going to put that back in this little bag. What we want, of course, is the bit with the DNA, which is here. So here is our DNA, all basically dissolved in some salt and detergent. Now I want to try and filter this, and I've seen on the web various people talk about various things to filter it with, but I found the best thing to do is to use ordinary kitchen roll. I've just put this into a little square, and I'm going to put it into a funnel, and I'm going to pour this in here. Now I've got quite a lot, so I've come prepared, and I've got another one which I'm going to do in the same way. So I folded up my kitchen roll, I open up one corner and it makes a little funnel, and then let's pour all this stuff in there. Yeah, it'll all go in. There we go. Right. So now, what do we need to do? Well, now we need to leave this experiment for a little time, because we want all of this strawberry juice to soak through the filter paper. Welcome back. For you, only a moment has passed, but for me, 2 hours have passed whilst this has been going through. So let's just take this out. And I've now got in both of these about 50 mils of strawberry DNA extract. So let's pour these together into one. And I don't want too many bubbles here. But with using washing up liquid, there's very little I can do about that. Now, I've got here some very cold isopropyl alcohol. It's just come out of the freezer. And I'm going to put in here about 2 to 3 centimeters depth of this very cold isopropyl alcohol. And you can see it's made a different sort of layer on top. And there's my very cold isopropyl alcohol. And now what I'm hoping is we're going to start getting a few bubbles, which I can see rise up through here. And what I'm hoping is we're starting to get in here some strands of DNA forming. And we should find that the DNA will prefer to be in the isopropyl alcohol layer rather than in this aqueous layer. Sometimes we can get it going a little bit more if we give it a little gentle stir. And I'll do that one. One of the reasons we use strawberries is because they have a lot more DNA in them than your average other plant. So here I've got the DNA. I can start to see it, especially in the overhead camera here. We can see this DNA forming, these little strands trying to glue together. You'll notice that I am stirring, but I am stirring really very gently, just trying to get these to mix a little bit, and the DNA coming out of solution of washing up liquid and salt and into the isopropyl alcohol. And now what I'm going to try to do is to see if I can actually pull a little bit of this out. And it's very sort of gloopy and difficult to get out, so I'll just try a pair of forceps here, long tweezers effectively. And I am getting out a little bit of DNA there. And there's some more. Quite hard to see when I put it down on the paper tissue. And it's quite difficult to get some of this to come up. As you can see, it's really quite small strands, but what we can do is perhaps wipe some of that off. And we've got here some DNA which is very sort of fine here, trying to pull this out. And we've got a few bits of DNA. So there we've managed to extract some DNA. Now, what's the sort of things that you're actually going to do with this. Well, for this is the end of this experiment, because what we were trying to do was just take out and extract some DNA. But in actual fact, once I've now got this DNA out, then I could do and perform various experiments on it and have a look at this DNA and see what sequences I've got. But all that is really beyond what we can do at home. So there you have extracting some DNA. You can just about see some of these very fine long strands as they clump together. Yeah. Extraction of DNA from a strawberry.